Right, the final video in the inheritance, variation and evolution chapter for the EDUQAS WJEC GCSE biology is all about the technique of genetic profiling, sometimes known as DNA fingerprinting, but don't get confused with the normal fingerprinting where people might leave the imprints from the fingers. They sometimes call it DNA fingerprinting simply because somebody's genetic profile is unique to that particular person, unless you have an identical twin. But let's go through the process of genetic profiling for you. It sounds quite a new technique, but this was developed in the 1980s, and there's a variety of applications that this can be used for. But you've got to bear in mind that every person has about 99.9% of DNA the same. That's 99.9%. However, 0.1% of 3 billion nucleotide bases is actually still quite a lot of differences. And that's what this technique uses. It tries to show the differences in the genome, differences in the genetic sequence, differences in the nucleotide bases from one person to another. How it does this is that chemicals called enzymes are used to cut DNA up in certain places of a person's genome. And so when you take somebody's genome and another person's genome, these enzymes should cut the DNA and in exactly the same place from one person to another. But what might vary from one person to another is the actual length of DNA fragments from one person to another may well vary. Technically, this is called VNTRs, or variable number tandem repeats. Sounds very technical, but what it is that some people have lots of nucleotide bases in the fragment, and another person may just have a few less or a few more, which means that bit of DNA fragment from one person will be longer than another person's. And that's what tends to be unique to every single person on this planet. Unless you've got an identical twin. So what then? You've got these DNA fragments. You can't see them. They're in test tubes and they're very, very small. So what people do is they stick these DNA fragments onto gel electrophoresis. It's a little bit like jelly. And when they apply an electric current, these DNA fragments start to separate. And the bigger pieces of DNA, the bigger fragments, take a bit longer to move through this gel, and the smaller fragments go fastest. So if you look at the picture now, you've got 1, 2, 3, 4, 5 bands, and the very bottom band, that's the smallest fragment of DNA, and the very top band is the biggest piece of DNA, the biggest fragment. You'll notice, however, that some of these lines, some of these bands are thicker than the others. That doesn't mean they're bigger pieces of DNA. What that means is that there's actually more than one fragment there of that particular size. So that very thick band, second from the bottom, that happens to be the second smallest DNA fragment, but there's lots of them there. So that's why it appears as a thick band. So as I said, this DNA pattern, this gene profile is completely unique to an individual, which means that if you have lots of other people's genetic profiles, the bands separate out like this, they can be compared to perhaps DNA found at a crime scene, a number of applications. So I've given you an example here of 3 people. I should have labelled A, B, C. Here's their genetic profile. You can see they're quite different from each other. But if we looked at a genetic profile of some DNA found at a crime scene, what we would see or hope to see is some similarity. Maybe not exactly perfect, but some similarity. And as this is coming up on the screen, you'll notice that person B here matches the crime scene genetic profile in terms of position of the bands and also the relative thickness of each band. As I said, there are a number of applications that this can be used for. Solving crimes, very useful in forensic science. It can also exonerate people. So if somebody's been falsely accused of a crime, sometimes it has led to people being released from prison. But it can also be used in paternity testing. This is quite commonly done. This is a case of determining if a man really is a father of a child. Sometimes blood test is not sufficient. So DNA testing, paternity testing, looking at the genetic profile of the children or child and the father will determine if that is the real father. The final picture I'd like to show you here is a real picture. This is called an autoradiograph, but it's a real picture of genetic profiles. It's taken of 7 suspects. It doesn't say that this has to be criminal suspects, but 7 people have had their genetic profiles looked at here. And you can see the banding patterns. Some are quite thick, some are quite thin, some are quite faint. And there's a blood stain in the middle. Just take a quick look at this picture and determine which suspect's genetic profile matches the blood stain in the middle.